Hi, Assalamualaikum and good day everyone. I hope you are in a very good health and conditions. Alright, on this video, I will discuss on chapter 5 titled Prokaryotic and Eukaryotic cells. By the way, my name is Madam Nurul Hidayah binti Wan Anuar and I will talk about on the first subtopic for this chapter. Okay, on this chapter, the first subtopic is on microscopy. Basically, there are two types of microscopes that will be discussed, which are the light microscope and also the electron microscope. Let's start it. Okay, so before we get into details of the microscope, should we first meet with the inventors who contributed to our invention today? So basically, microscopes are a major tool in cytology, which is the study of the cell structures, because the cells are very small to be seen by our naked eyes. Alright, so this handsome guy here was an eyeglass maker named Zacharias Janssen, and Hans was his father. This Hans and Zacharias Janssen created the first compound microscope in the 1590s, when there was at that time where the magnifying glasses was introduced with 5-time magnification. Okay, so afterwards, Anthony van Leeuwenhoek and Robert Hooke made improvements by working on the lenses. Basically, Robert Hooke was among the first to make significant improvement to the basic design, where he is the first person to record and observe the cells. Basically, in his study, he see the non-living particles, which they basically invent the term cellas, which refer to the open spaces after viewing the slices of cork through a very primitive microscope. Hooke microscope share the common feature with the early telescope, and Anton van Leeuwenhoek afterwards made further improvements when he achieved to magnifying power up to 266 times larger than actual size samples using only a single lens. Basically, he used to look at everything from rainwater to tears, and he saw a swimming living cells, which is the microbes. At that time, he called the microbes as animalcules, but perhaps the animalcules were the microorganisms, or the protozoans, and also the red blood cells, as well as the spermatozoas that is found in his sample. Okay, so now let's take a look at the principles of microscopy. Okay, so first of all, the discovery and early study of cells progressed with the invention and improvement of microscopes in the 17th century, which leads to the inventions of the light microscope. Alright, so for important parameters in microscopy are magnification and resolving power, also for the resolutions. Okay, so what is magnification? Basically, the magnification is the ratio of an object's image to its real size. And resolving power, in the other hand, is the ability of the lens to show two adjacent objects as discrete. This is very important, where the smallest or minimum distance of 2 points can be separated and still can be viewed as the two separated points. And last but not least, the parameter that is important is the resolution. This resolution indicates the level of details actually observed in the specimen. It is limited by the wavelength of the source, and in this case of the microscope, the wavelength of the source is the light. Okay, it follows the formula of the smaller the wavelength, the smaller the object that will cause the diffraction, the better the resolution, the better the resolving power. Okay, so let's discuss on the parts of the microscope. So the structural element of the microscope consists of the head, okay, and the arm that connect the head to the base. Okay, so this base is where the microscope stands on. Okay, so next is the eyepiece, which also refer to the ocular. So the eyepiece is the lens nearest to your eyes. Okay, so this is the interpupillary adjustment to adjust the distance of the eyepiece. Okay, this is the nose piece, the circular structure where the different objective lens are screwed in. Okay, to change the magnification power, so basically you just simply rotate the turret. Okay, so these are the objective lenses that usually you will find 4 lenses on the microscope. The shortest lens is 4 times, followed by 10 times and 40 times, and the longest lens is the 100 times. Okay, so this is the mechanical stage, which is the platform to support the slides. And this is the stage controls that usually have the 2 knobs. One knob moves the slide to the left and the right, and the other one will move it forward and backwards. Okay, so this is the diaphragm. Okay, it is located under the stage, which can be adjusted to vary with the intensity and size of the cone of light projected through the slides. And this is the illuminator, where a steady light source that shines up through the slide. And this is the coarse knob to bring the specimen into focus. And this is the fine knob used to fine-tune the focus of a specimen in conjunction with the coarse knob. Okay, so this is the switch, the light switch to switch on the lights, and last but not least is the brightness adjustment, where you can adjust the brightness so it is suitable for your vision. Okay, so basically the visible light rays will passes through the specimens, and it will uses the glass lenses to view the objects. Okay, so the different magnifications of the objective lenses ranging from 4 times, 10 times, 40 times and 100 times. Basically, the lenses of 4 times to 40 times are the dry lenses, and the 100 times is the wet lenses, which we should use it with the oil immersion. For the magnification power, we can achieve it by multiplying the objective lens with the ocular lens. It means when you are observing a specimen under the 4 times magnification power for the objective lens, you should multiply with the power of the ocular lens, normally 10 times. So you should get 40 times total magnifications for the specimen. Okay, so now let's discuss with the first microscope, which is the light microscope, or also known as the compound microscope. Alright, so light microscopes basically can magnify effectively to 1000 times the size of the actual specimen. And at the higher magnifications, the image basically will blurs, so you need to adjust the fine coarse to get into the fine focus, or you need to use the higher magnification lens, higher objective lens, which is 100 times, and you need to put the oil immersion. Okay, so the light microscope can resolve individual cells, but it cannot resolve much of the internal anatomy, especially the organelles. Okay, so let's see the size of measurements of the images that we can see with our naked eyes versus the microscope. Okay, so basically, our naked eyes can only see the specimen up to 100 micrometer. Okay, so basically, we can observe the hair that measurement of the sizes is ranging from 60 to 100 micrometer. Okay, but we can improve our vision with using the optical microscope, or also known as the compound or light microscope, that we can use it to observe the smaller specimens such as the bacteria, such as the lactobacillus, which is the sizes is about 1 to 50 micrometer. Other than that, we can use the more advanced microscope, which is the electron microscope, that can be magnify the images of the smallest specimen up to 1 nanometer, so that we can use the electron microscope to see the DNA. This is the other type of the light microscope, called as the stereo or dissecting microscope. So the stereo or dissecting microscope allow the user to see the 3D structure, and it is very useful to be used for surgery and dissection. And this is the image showing the microscope, and as you can see, it is very well made, and you also have the adjustable magnifying system here. All right. Okay, so this one is very good for studying overall structures of insects and the leaves, and also it is used to study the larger cellular structure, but not the organelle. If you want to observe the organelles, you need to use the electron microscope. Okay, as I mentioned earlier, to resolve a smaller structure, we can use the electron microscope. So this electron microscope focuses a beam of electron through the specimen or onto its surfaces. It depends on the types of the electron microscope. Okay, because of the resolution is inversely related to the wavelength, so the electron microscope with shorter wavelength than the visible light will have finer resolution. Okay, so theoretically, the resolution of a modern electron microscope could reach 0.1 nanometer, but the practical limit is closer to 2 nanometers. There are two types of electron microscope, which are transmission electron microscope, also known as TEM, and the second one is the scanning electron microscope, also known as the SEM. Okay, so let's discuss on the first type of the electron microscope, which is the transmission electron microscope, or TEM. Okay, so for the TEM, it is a same design as the light microscope, just only that the lenses are electromagnets. Okay, so this microscope passes the electrons, not the lights, okay, through a specimen, and it will focuses with the magnetic lenses, and it will project image onto a fluorescent screen or a photographic film, and it will produce a detailed image in static, 2D, and highly processed. So these are the image that you can get by using the electron microscope. Okay, using the TEM, you can see this image in a 2D, and you also can see this image in a very detailed manner. Okay, so this is the examples of images that we can observe using the electron microscope, particularly the TEM. Okay, so by using the TEM, we can observe the smaller objects, for example, the bacteria, and we can see in a very detailed manners. For example, here the Salmonella typhimurium and the Escherichia coli, examples of the bacteria. Okay, you can see the shape of the bacteria, which is bacillus, and also we can see the detailed structure of the bacteria also. And other than that, using the TEM, we can also observe smaller structure, for example, the chloroplast, which is the organelle. Okay, so here you can produce the 2D structure of the chloroplast, as you also can observe the inner structure of the chloroplast. The second type of the electron microscope is the scanning electron microscope, or SEM. Okay, for this scanning electron microscope, the electron is passes over the surface of a metal coated specimen, and it will be collected to produce a television type 3D pictures on a screen. Okay, so for this one, it is only provide the topographical info on the surfaces of the cells and tissue. Okay, so you can see the structure for the SEM is more likely to have the 3D structure, as you can see the real structure of the red blood cells here, as well as the sickle cell. Okay, here are another examples of the images captured by SEM, where you can see the dust mite among the skin scales here. So you can see the 3D structure of a detailed structure of a dust mite. Okay, so this is more examples of the images taken by SEM. As you can see, all the images here is in the 3D structure, and it only provides the topographical image that focuses on the surfaces of the specimen. So here, for example, this is the cancer cells taken by the SEM, and this is the paramecium, a ciliated protozoa. As you can see, you can observe the cilia of these protozoa. And this is the tongue bacteria that we can get from our tongue. Okay, so this is the image comparison for the SEM image and the TEM image. As you can see here, for the SEM image, you can see this image in the 3D structure, and for the TEM image, you can see this is in the 2D. For the SEM image, you can see this is the topographical structure that it focuses on the surfaces of the specimen only. But for the TEM, the electron can go beyond the surfaces and go deep into the specimen, where you can see the inside of the specimens also. Okay, so this is the comparison between the three types of the microscope here. The first one is the light microscope. These two are the electron microscope, the TEM, and also the SEM. Okay, so how it works is basically on the placement of the specimen. As you can see here, for the placement of the light microscope, the specimen is placed just below the condenser, as well as the TEM. Okay, and for the SEM, the specimen is located at the bottom side of the microscope. Other than that, the differences between these three microscopes is that on the sources of the wavelength. So for the light microscope, it used the light, whereby for the TEM and the SEM, it used the electron. Just that for the electron in the TEM is passed directly towards the specimen, and for the SEM, first it will scatter, okay, and it will scatter by this scanning coil, then it will be collected in the 3D dimensional specimen. Okay, last but not least, we are going to discuss on the differences between the SEM and TEM. So basically, for the TEM, the electron beam will passes through a thin samples and it is direct. Okay, and for the SEM, the electron beam scans or scattered over the surfaces of the samples, and the samples can be in a thick sizes. All right, and for the TEM, usually we prepare in the in the thin samples. Okay, and for the SEM, it can be in any thickness, and it will be mounted on the aluminium stub. All right, and the placement of the TEM, the specimen in this TEM, the placement of the specimen is halfway down the column, whereby in the SEM, the specimen is located at the bottom of the column. All right, and other than that is that the image will be shown on the fluorescent screen, and for the SEM, the image will be shown on the TV monitor. Okay, last but not least is about the images that is produced by the TEM is the 2D projections, and for the image that is produced by the SEM is the 3D of a specimen. Okay, so that's the end of the first subtopic of chapter 5, the prokaryotic and eukaryotic cells. So by then, see you again.